Hello and welcome to this session. In this session, we are looking at the principles of human genetics. Guys, principles of human genetics are rules or standards that normally govern the biological phenomena of heredity. Remember, heredity is the transmission of characteristics from parents to offspring via DNA. Okay, and these units of transfer, we call them the genes. Okay, so these are the principles that we are going to study in this session. Welcome to the session. So by the end of this presentation, you should be able to describe cell specialization. You should be able to describe body tissues, the four body tissues, describe the process of cell-to-cell communication, describe the human genetics, lastly describe the process of induction of a cell. So those are 5 objectives. Okay, the first one, specialization. And cell specialization is simply the ability of each part of a cell to perform specific activity and then integrate those activities with other cells for harmonious functioning of the whole body of the organism. Remember, each particular part of a cell, any organelle, has to play a distinctive and a specific role. If it's mitochondria, respiration, centriole, cell division, ribosomes, protein synthesis, each organelle has to play its particular role. Remember, playing this role is for the overall welfare, and it's is in general, and also is very important for the entire body. So the cells need other cells that are, although different in structure, so that they can work towards and work in a concerted effort to undertake a specific function at a higher level. So the different types of organelles need each other for us to achieve these functions. So we have different parts of a cell or the cell organelles, starting with the cell membrane or the plasma membrane. This is the outer boundary of a cell that normally regulates the passage of substances in and out of the cell through processes such as diffusion, active transport, pinocytosis, phagocytosis, and so on. This cell membrane provides protections and maintains the cell integrity. Okay, remember this human cell lacks a cell wall, so cell membrane normally plays the role of the protecting the other cells. Next, we have the cytoplasm, and the cytoplasm, this is gel-like substance that normally fills the cell containing organelles, enzymes, various molecules required for cellular processes. The nucleus, this is the cell, the central control center of a cell. It contains the DNA, the genetic material, and it directs cellular activities such as protein synthesis. Mitochondria is the next one, and mitochondria are organelles responsible for production of energy. We call it ATP, okay? So through cellular respiration, they are commonly referred as the powerhouses of a cell. Endoplasmic reticulum, abbreviated as ER, is a network of membranes involved in protein and lipid synthesis. So we have the rough endoplasmic reticulum, which has ribosomes attached to it, while the smooth endoplasmic reticulum lacks the ribosomes. The ribosomes are simply cellular structures where protein synthesis normally occurs. They are either attached to the rough endoplasmic reticulum or free in the cytoplasm. Golgi apparatus or the Golgi complex, these are stack of flattened membranes that processes, modifies, and packages proteins and lipids for transport within and outside the cell. Next, we have lysosomes. And what are lysosomes, guys? They are simply organelles that contain digestive enzymes that normally breaks down waste materials, cellular debris, and foreign invaders. They help with cell recycling. Also, the vacuoles, these are membrane-bound sacs that store water, nutrients, and waste products. So we have smaller or less prominent in human cells compared to plant cells where they are in abundance. Cytoskeleton is a network of protein filaments, e.g. the microtubules and the microfilaments, and these ones, they are responsible for maintaining the cell shape, aid in cell division, and also enables cell movement. Lastly, we have the centrosome. This contains centrioles, and centrioles are involved in cell division and organization of microtubules during cell division. Remember, the centrioles, they normally duplicate during the interphase, the G1 phase. So where are we, guys? We have already described what cell specialization is. It is high time now go to describing body tissues, and we are saying that they are four, okay? Four body tissues. We have the connective tissue, epithelial tissue, muscle tissue, and nerve tissue. We abbreviate it as ECMN, epithelial, connective, muscular, and nerve tissue. So starting with the by definition is that body tissues are a group of similar cells that work together to perform specific function in a body. Remember, cells make up tissues, okay, and tissues will be making up organs will be making up organ systems, and the organ systems are going to make up an organism, a complex structure like all of us. So epithelial tissue, I find that epithelial tissue are composed of closely packed cells that form lining or covering of various body surfaces and cavities. Their major role is protection, so they serve as protective. Sometimes in the GIT you find they have absorptive role, secretory, and sensory functions. Examples include the skin epidermis, the lining of the GIT tract, and the respiratory tract epithelium. Connective tissues, these are characterized by cells dispersed within the extracellular matrix that includes fibers and ground substance. These tissues provide structural support, connect different body parts, and store energy and play roles in immune responses. Examples of connective tissue will be including the bones. The bones are connective tissue, cartilage, adipose tissues, blood, and the lymphoid. We have the muscle tissue as number 3, and the muscle tissue consists of specialized cells called the muscle fibers that have the ability to contract to generate force and movement. So we have 3 types of muscle tissue. The skeletal muscles, these are responsible for voluntary movements and are normally attached to bones. We have smooth muscles. These are found in the walls of organs and structures like the digestive tract and blood vessels, controlling involuntary movements. We have the cardiac muscles, and these are exclusive to the heart and responsible for pumping blood. Lastly, the fourth connective tissue is nerve tissue, and nervous tissue is composed of neurons. Okay, neurons, these are nerve cells, and also glial cells, these are supportive nerve cells. So neurons transmit electrical signals and process informations, allowing communications within the nervous systems, while glial cells do support and protect the neurons. Remember, a link will be popping up on the different types of glial cells found in the human body. You can review to see which ones are found in the central nervous system and which ones are found in the peripheral nervous system. So if you try to describe, and starting with the epithelial tissue, this is one of the simplest and basic structure through which individual cells make the foundation of building up tissues of varying complexity. Epithelium is a living tissue, and in its simplest form, it consists of a single layer of cells covering the surface of the body and organs within it. Epithelial tissues normally line various spaces and tubes in which they are referred to as endothelium. So typically, individual cells firmly attach to each other and rest on a basement membrane and have a free surface. The main function of epithelial tissue is protection. So we have the nomenclature of epithelial tissue, epithelial cells. So how are they named? We're saying that one of the major characteristics that we use is shape. So we say we have squamous epithelial cells. So how do we arrive at squamous epithelial cells? We say squamous because cells are flat, thin, and have a scale-like appearance. They are often forming a single layer and are well suited for functions like diffusion and filtration. Number second class under shape is cuboidal epithelials. Right from the cube, they are cube shaped. So you find that approximately they have equal height, width, and depth. They are commonly found in the glandular tissue and areas where secretion and absorption is occurring. Number third, according to shape, we have columnar epithelium. So these cells are tall and rectangular with their height being greater than their width. You find that they are found in the lining of the digestive tract and often have specialized functions like secretions and absorption. Another characteristic that we use to name epithelial cells is layering, or the number of layers. So simple epithelium, in simple epithelium you find that there is only one layer of cells, typically found in areas where we have absorption, secretions, filtration. Examples, you can have simple squamous lining the blood vessels. So simple meaning the number of layers is one, squamous meaning they are flat cells, thin cells. Okay, so simple squamous. What about stratified epithelium? This type consists of multiple layers of cells stacked on top of each other. They provide protections and are commonly found in areas exposed to wear and tear, such as the skin, where we have stratified squamous epithelium. So they are made up of multiple layers and they are flat cells. We also have specialized types like the pseudo-stratified. Pseudo stands for or denotes what? False. So pseudo-stratified, false layers. So although it appears stratified, all cells in this particular type of epithelium are in contact with the basement membrane. So it's typically found in the respiratory tract where it may have the cilia for moving mucus. Okay? Another specialized type of epithelium is the transitional epithelium. So this specialized type of epithelium is found in the urinary system and particularly in the bladder. It has the ability to stretch and accommodate changes in volume. Glandular epithelium is the other one. So we have endocrine glandular epithelium and exocrine glandular epithelium. In endocrine glandular epithelium, this type of epithelium forms endocrine glands that normally secrete hormones directly into the bloodstream. These glands lack ducts. Whereas exocrine glandular epithelium, exocrine glands secrete their products through ducts into the body surfaces or cavities. Examples could include sweat glands or salivary glands. Very good. We also have another criteria to classify epithelium where we talk of cilia. So some types of epithelium cells are equipped with hair-like structures, the one that we refer to as cilia. And so for those who have cilia, we normally refer to them as ciliated epithelial cells. And they are involved in moving mucus and other substances along surface of the tissues. So we could have them in the respiratory tract. We could also have some ciliated in the fallopian tube. Okay, so the coordinated beating of the cilia helps transport materials in a specific direction. Mucus for the respiratory and the sperms, okay, for the fallopian tube. Okay, we have ciliated epithelium. which is common found in respiratory tract, where it can help move mucus that is being trapped and pathogens out of the way. And that's just an example of the ciliated. So we could have ciliated or non-ciliated. One other characteristic is keratinization. Keratin is a protein. So this is the process by which certain epithelial cells produce and accumulate the protein keratin. Remember, keratin is a tough fibrous protein that normally provides protection and waterproofing to the cells. So epithelial cells that keratinize are typically found in the skin, specifically in the epidermis. So these cells are known as keratinocytes. So the keratinization process results in the formation of a tough, durable layer of the skin that is resistant to mechanical damage and helps prevent water loss from the body. So the different types of epithelial tissues we could have simple squamous epithelium. This one is found in the lining of blood vessels, that is the endothelium, and in the alveoli of the lungs. Also in the kidney glomeruli, you'll find the simple squamous epithelium. Simple cuboidal epithelium, this will be found in the kidney tubules and in the glands and ducts, e.g. the salivary glands. We have simple columnar epithelium. This one normally lines the digestive tract, especially the stomach, small and large intestines. Okay. It also lines the uterus and uterine tubes. Pseudostratified columnar. This is found in the respiratory tract, especially the trachea and the bronchi. Okay, we also have portions of male reproductive tract, that is the epididymis, where we find the pseudostratified. We also have stratified squamous epithelium, and stratified squamous epithelium you find in the epidermis of the skin. Most of the time you find it is keratinized, okay, the outer layer. It also lines the oral cavity, esophagus, and the vagina. Stratified squamous epithelium. Okay. Stratified cuboidal epithelium, it's rare but is found in some sweat and mammary gland ducts. Stratified columnar, it's rare but is found in some areas of the male urethra and large ducts of some glands, whereas transitional epithelium lines the urinary bladder, ureters, and parts of the urethra. The next tissue is the connective tissue. This is one of also the primary tissues in the human body and is characterized by diverse and specialized cells. Consists of the extracellular matrix and plays a number of functions. So the extracellular matrix contains the ground substance, protein fibers, that is elastic fibers, collagen fibers, and reticular fibers. We also have resident cells, the mesenchymal cells, macrophages, adipocytes, and the fibroblast. Major characteristics of the connective tissue, they have an extracellular matrix, and this extracellular matrix is a complex mixture of proteins and carbohydrates that surrounds and support the cells within the connective tissue. Remember, we also have cellular components, and a good example is the fibroblast. I'll be showing you the different types of cells in the connective section. So the extracellular matrix of both loose and dense connective tissue proper is composed of this amorphous ground substance, extracellular fluid, and collagen, elastic, and reticular fibers. So we can have the ground substance, the material that is in the space around cells. It largely consists of proteins and fluids, whereas the fiber, we have the collagen fibers, elastic fibers, and reticular fibers. So reticular fibers are made up of collagen, thin branching, whereas elastin are made up of elastin. These are rubber-like protein, thinning and branching. Collagen fibers are made up of collagen, and this is a fibrous protein. It is the most abundant and the strongest of the three. Okay? Correct, guys. Next, we have this types of extracellular components. The fibrous proteins. It talks of the collagen and elastin. Okay, collagen is the most abundant protein in the extracellular matrix. It provides tensile strength and structural integrity to tissues, while elastin fibers give tissues elasticity, the ability to stretch and recoil. Yeah, protoglycans are coproteins and long chain of carbohydrates. So they are called glycoaminoglycans, the GAGs. Okay, so these molecules form complexes with water, creating a gel-like substance in the extracellular matrix. One of the GAGs is the hyaluronic acid. It's a type of a GAG that is particularly important in providing viscosity and lubrication in the extracellular matrix. Another major component is the glycoproteins, and these are just proteins that are attached to carbohydrate chains. They play a role in cell adhesion, signaling, and tissue organization. One of the best examples we have fibronectin, which helps cells adhere to the extracellular matrix and facilitates migration. Laminin is another example of glycoprotein found in the extracellular matrix. This one forms the basis for the basement membrane, and it aids in cell attachment and tissue organization. Water is another component of the extracellular matrix. So water molecules are essential in maintaining the hydrated gel-like consistent of extracellular matrix, which is crucial for the mechanical properties and cell function. So a number of cells found here, examples of connective tissue cells. We have fibroblasts. This one they normally produce and maintain extracellular matrix. They are elongated and spindle-like shaped. Adipocytes, they store energy, regulate metabolism, provide cushioning, contain a single large lipid droplet. Macrophages, these are immune response, phagocytosis, tissue repair. So they are important in those functions and they vary in shapes. Okay, they are phagocytic, they eat the debris. Yeah, mast cells, they are important in immune response, inflammation, and allergic reactions. They are normally round or oval shaped and they contain granules. Plasma cells, these are antibody production in the body. Okay, and they are eccentric, they have an eccentric nucleus and specialized for antibody production. Okay, antibody G, A, D, M, and E. Guard me. Okay, guard me. G, immunoglobulin G, immunoglobulin A, immunoglobulin D, immunoglobulin M, immunoglobulin E. Chondrocytes, these normally maintain the cartilage matrix and they are located in the lacuna within the cartilage matrix. Osteoblasts and osteocytes, these are bone forming and maintaining cells, okay, along with osteoclasts, okay, and other osteogenic cells. So osteoblasts lay down bone matrix, osteocytes are embedded in the bone matrix. Another example for this matter, we can end with the pericytes. This one, they regulate blood flow, vessel stability, and tissue repair, and they have contractile cells along the capillary walls. So what are the functions of the connective tissue? Connective tissue is involved in providing structural support and maintaining shape, attaching muscles to bones, those are tendons, or attaching bones to bones, the ligaments, or facilitating nutrients, gas, and waste product transport. They also participate in the body's immune response and they help in storing energy by use of the adipose tissue. They are also important in repair and regeneration of tissues. We have the types of connective tissue, where we have loose connective tissue. Loose connective tissue is characterized by loose arrangement of the fibers and cells within the semi-fluid ground substance. It provides support and flexibility, and it's normally found beneath the skin, around blood vessels, and between organs. Loose connective tissues include areola, adipose, and reticular tissues. Dense connective tissue is composed of densely packed collagen fibers, making it hard and resistant to stretching. So dense regular connective tissue, and we have also dense irregular. So dense regular connective tissue has collagen fibers that are arranged in parallel bundles, providing strength and stability. It forms tendons and ligaments, dense regular. Whereas dense irregular, it consists of collagen fibers that are irregularly arranged, making it suitable for resisting multidirectional stress. It is found in the dermis of the skin and capsules of the organs. We also have specialized connective tissue. Specialized connective tissue include the cartilage, where we have hyaline cartilage found in the nose and the trachea. We have the elastic cartilage found in the ear and the fibrocartilage found in the intervertebral disc. Another specialized connective tissue is the bones or the osseous tissue. Osseous tissue consists of two major parts. We have the compact bone, okay, that is dense and strong, and the spongy bone that is porous with trabeculae. Blood is also another specialized connective tissue, and it's unique fluid connective tissue that normally circulates throughout the body. It consists of the liquid part, which is the plasma, and the formed elements that consist of erythrocytes, leukocytes, and thrombocytes. So the work of blood is to transport oxygen, nutrients, hormone, and waste products, while also playing a critical role in immune responses and clotting. Lymphoid tissue is another specialized connective tissue, and it consists of the lymph nodes, the tonsils, and the spleen. It is specialized for immune response, filtering lymph, and producing immune cells. Connective tissue is our third tissue, and this is a specialized type of tissue found in the human body that forms the basis for the nervous system. It includes the brain, spinal cord, and peripheral nerves. It normally plays a crucial role in transmitting electrical signals such as nerve impulses or action potential through the body to control and coordinate various physiological processes. Nervous tissue is normally composed of two primary cells, the neurons, which are the nerve cells, and the glial cells or the neuroglia, which are the supportive nerve cells. So the neurons are the fundamental units of the nervous system and responsible for transmitting those electrical signals. They consist of cell body, which is referred to as the soma, the dendrites, okay, and the axons. So neurons communicate with each other and with the target cells. They could be muscles or the glands through what we call synapses. So these are just specialized junctions where neurotransmitters are released to transmit signals. Glial cells, these are non-neural cells that provide support, protection, maintenance of the neurons. They outnumber the neurons in the nervous system. Okay. Most types of the glial cells include the astrocytes, the one that is used to maintain the blood-brain barrier, oligodendrocytes in the central nervous system, and the Schwann cells in the peripheral nervous system. These ones are involved in production of the myelin sheath that normally wraps around axon and enhances the speed of nerve impulse transmission. This will be affected in conditions such as multiple sclerosis and Guillain-Barré syndrome. Microglia are immune cells of the nervous system. They act as phagocytes. They engulf and remove cellular debris and pathogens and damage neurons. We have ependymal cells. These cells they line the cavities of the brain and the spinal cord and produce cerebrospinal fluid, which cushions and protects the central nervous system. So guys, which glial cells is involved in the production of the cerebrospinal fluid? Epidermal cells, very good. Functions of the nervous tissue include sensory perception, integration, motor output, and communication. This is an example of a nerve tissue: the dendrites, cell body, nucleus, the axons. You can see the myelin sheath, the Schwann cells, the rod of Ranvier, the junctions, and then the terminal axon. Lastly, we have the muscle, and the muscle is the last of the 4 primary tissues in the body, along with what we have already discussed: the epithelial tissue, nervous, and connective tissue. Muscles are normally specialized for contraction, generating force, and movement. We have 3 types of muscle tissues in the body. These muscle tissues functions by contracting when stimulated by electrical signal. So this contraction generates force and movement, enabling various functions from simple reflexes to complex activities like walking and maintaining a heartbeat. So the different types of muscles have distinct properties and roles in the body, but all contribute to the overall function and the mobility of the human organism. So you have the skeletal muscles, which are also known as striated or voluntary muscles. So they are striated because we have, when you look at under microscope, you will see contractile proteins. Okay, they are multinucleated, so they have several nuclei per cell. They are voluntary. Skeletal muscles normally act under conscious control. You are aware of what is happening, allowing individuals to initiate and control movements intentionally. We only have one exception, and this is the diaphragm muscles that normally works non-involuntary. Skeletal muscles is also involved in activities such as walking, running, jumping, and lifting items. Smooth muscles are non-striated, and also they are involuntary muscles. They are majorly found along the walls of the internal organs, blood vessels, and other structures. So they lack striations, okay? So they are non-striated. They are unicleated. So it means they have a single nucleus per cell as opposed to the skeletal muscles. They are involuntary in nature. Okay, the smooth muscles are not under conscious control. They contract involuntarily. This could be in response to hormones, nerve signals, or stretching of the tissues. Smooth muscles play a crucial role in the contraction of organs like the digestive tract, blood vessels, uterus during childbirth. Lastly, we have cardiac muscles. So these are they are just exclusive to the heart and responsible for pumping blood throughout the circulatory system. They are striated like the skeletal muscles, okay, so they exhibit a striated appearance. They are uninucleated like smooth muscles, except for them they could be having 1 or 2 per cell. They are involuntary in nature, just like the smooth muscles, okay, so but they can be influenced by the autonomic nervous system. Cardiac muscle normally contract rhythmically to pump blood, and its unique properties to allow coordinated and synchronized contraction. Okay, so we have different types of body systems and the organs and the roles. Yeah, the integumental system consists of the hair, skin. Skeletal consists of the bones, skeletal, and cartilage as the organs. Muscular system, we have the skeletal muscles as organs. Nervous system, we have the brain, spinal cord, nerves as the organs. Endocrine system consists of the organs like the pituitary gland, thyroid gland, etc. Cardiovascular system consists of the heart, blood vessels, blood. We have the lymphatic that consists of the lymph node, the spleen, and the thymus. Respiratory, we'll be looking at the bronchial tree, together with the lungs, trachea, and the bronchi. digestive system, stomach, liver, small intestines, large intestines, pancreas, etc. Urinary system consists of the coop, kidneys, ureters, bladder, urethra. Reproductive system consists of the testes, penis, prostate, just for the male, and also the immune system consists of the lymphocyte, white blood cells, and other tonsils. So these are some of the functions. Remember, a link will be popping up on the 12 body systems outlining the function of each system. So guys, where are we? We are done with describing cell specialization, describing body tissues. Now we can look at describing the process of cell-to-cell communication. So what is cell-to-cell communication in human cells? This is a complex and highly regulated process. It allows individual cells to coordinate their activities, to respond to changes in the environment, contribute to functioning of tissues, organs, and the entire organism. This communication normally occurs through various signaling mechanisms, which can be broadly categorized into two. Could have direct or indirect chemical communication. So on direct communication, we normally have gap junctions. So gap junctions, these are specialized channels formed by protein called connexons. So these channels create pores and tunnels that directly connect the cytoplasm of adjacent cells. So this gap junction normally allow for direct exchange of ions, small molecules like nutrients, metabolites, and signaling molecules, e.g. the cyclic AMP, inositol triphosphate, between these neighboring cells. They play a crucial role in coordinating the activities of cells within tissues. For example, in the cardiac muscle tissues, you find that gap junctions ensure synchronous contraction of adjacent heart muscles, okay, allowing the heart to pump effectively. They enable the rapid spread of electrical impulse and biochemical signals, ensuring that cells act in a coordinated manner. Remember, gap junctions is just but one of the cell junction. Okay, a link will be popping up and you can be able to see the other types of cell junctions. In indirect cell communication, we're saying that we could have endocrine signaling. So this involves the release of hormones into the bloodstream by specialized endocrine glands. These hormones travel throughout the body and bind to specific receptors on target cells, triggering various responses. You could have an example of insulin that is normally released by the islets of Langerhans, the beta cells, and they go to act on different cells, target cells, so as to improve the uptake of glucose, regulating the blood glucose levels. Paracrine signaling, okay, So in paracrine signaling, cells release signalings that we refer to them as paracrine factors into the extracellular fluid, which affect nearby target cells. These signaling molecules include growth factors and neurotransmitters. For instance, we have nerve cells. They normally release neurotransmitters at synapses to transmit signals to adjacent neurons or muscle cells. We could have neurotransmitters such as acetylcholine, glutamate, okay? So a link will be popping up to see the most common 20 neurotransmitters in the body. We also have autocrine signaling, and this one normally occurs when a cell releases signaling molecules that bind to receptors on its own surface. So this type of signaling is involved in cell growth and regulation. Cancer cells often exhibit this dysregulated autocrine signaling. They signal themselves. Okay. So neurotransmission in the nervous system, neurons communicate through the release of neurotransmitters at junctions. So when an action potential reaches the end of a neuron, okay, neurotransmitters are released in the synaptic cleft where they bind to receptors on the postsynaptic neuron or target cell, transmitting signals. Immune signaling, cells of the immune system such as T cells and B cells communicate through the release of cytokines and other signaling molecules. These molecules help regulate immune responses, process of inflammation, and immune cell activation. Chemical signaling in cell growth and differentiation. We find that signaling pathways play a critical role in cell growth and differentiation during development and tissue repair. So guys, that's the end of the cell-to-cell communication. We can now look at describing what human genetics is. So genetics is a field of biology that studies the inheritance, variation, and expression of genes in humans. Definition of terms, we could have genetics, which is the scientific study of heredity and variation of inherited characteristics, whereas genome is the complete set of individuals' genetic material, including their genes and non-coding sequences of the DNA. Remember, a gene is a segment of a DNA that contains instructions for making specific proteins or set of proteins, which in turn determines an individual's traits. An allele is a variant form of a gene that can result in different traits or characteristics when present at the same genetic locus. Genotype, this is the specific genetic makeup of an individual, typically referring to the combination of alleles at a particular gene or set of genes. What about phenotype? Phenotype are observable physical or biochemical characteristics of an organism. They may result from interaction between its genotype and environment. What's the difference between homozygous and heterozygous? So in homozygous, an individual has two identical alleles at a specific gene locus, like AA, capital A, small a. So heterozygous is when an individual has two different alleles at a specific gene locus. So we have capital A, small a. So when do we say it's dominant? When do you say it's recessive? It is dominant when an allele that is present will express its traits in phenotype, even if the individual has one copy of the allele. Okay, it may be this, but still someone is showing the trait, or this other one, but still someone is showing the trait. Okay. Recessive refers to when an allele that is expressed in phenotype only when an individual has two copies of it. Okay, it is not masked by a dominant allele. Okay, so we also have some other terms like a pedigree. Pedigree is simply a graphical representation of genetic relationship within a family over several generations. It's often used to study the inheritance and traits and diseases. Mutation is a permanent alteration in the DNA sequence of a gene, which can lead to changes in protein function and potentially result into genetic disorders. Genetic variation is the diversity of alleles and genetic traits within a population. What is genetic inheritance? The process by which genetic information is passed from one generation to another, whereas a karyotype is a visual representation of individual chromosomes, usually arranged by size and shape, used to detect any chromosomal abnormality. Genetic disorders is a condition caused by abnormalities in the individual's DNA, often resulting in physical or physiological abnormality or diseases. While genetic counseling, this is a medical and counseling service that normally helps individuals and families understand the genetic basis of inherited conditions and helps them make informed decisions about their health and reproduction. Genome sequencing is the process of determining complete DNA sequence of an individual's genome, providing a comprehensive view of their genetic makeup. While gene editing is the technique of modifying an individual's DNA, typically using the tools like the CRISPR-Cas9 to correct any genetic defects or introduce desired changes. So we have the central dogma of molecular biology, which is simply a fundamental concept that describes the flow of information within human cells. Okay, it describes how genetic information flows from the DNA to the RNA, this through transcription, and then from RNA to proteins, the one that we refer to as translation. It is essential for the synthesis of many proteins and molecules necessary for structure and functions of human cells. This process consists of 3 steps. We have first one, replication. So during DNA replication, the DNA molecules is duplicated to create a one copy, and this one normally occurs before cell division in the interphase, the S phase to be particular. It ensures each daughter cell receives a complete set of genetic information, whereas transcription is a specific segment of DNA is used as a template to synthesize a molecule called the messenger RNA by the aid of the RNA polymerase. So this reads the DNA code and creates a complementary messenger RNA. So this messenger RNA contains genetic instruction needed to build a protein. Then translation comes in, so whereby, of course, we are saying this is the final stage of the central dogma, and it takes place in the ribosomes, where the transfer RNA molecules reads the messenger RNA and brings the corresponding amino acids to the ribosomes. Remember, amino acids are the building blocks of proteins. So the ribosomes assembles these amino acids in a correct sequence to form a protein according to the instruction encoded in the messenger RNA. So guys, what do you understand by the term chromatin? Chromatin is found within the cell nucleus. It is the membrane-bound organelle that normally houses the cell genetic material. It serves as the relaxed extended form of the DNA within the nucleus during most phases of the cell cycle. In the interphase case, we normally have the chromatin, okay? So they play a crucial role in packaging, organizing the DNA, making it accessible for various cellular processes such as transcription and replication. How is it related to the DNA? You find the chromatin is composed of the DNA wrapped around histone proteins. It presents the primary structural form of the DNA to the nucleus, whereas the gene are specific segments of the DNA that are found within the chromatin, which in turn is located inside the nucleus of most eukaryotic cells, of which human cell is one of them. So genes contains genetic instructions for making specific proteins or the functional RNA molecules. They are responsible for encoding traits and function in an organism. Genes are a sequence of DNA. Within the chromatin, DNA regions are corresponding to genes that are actively transcribed into messenger RNA during gene expression. What about chromosomes? What are chromosomes? Chromosomes are found inside the nucleus of eukaryotic cells, particularly during cell division. In mitosis, we find it is during the anaphase stage. That's where we are able to see the chromosomes. Okay, remember in prophase, the chromatin are beginning to condense, okay, and thicken so that they are visible. Chromosomes are also present in a less condensed form as chromatid during interphase. So chromosomes are highly compacted and organized structure of the DNA and proteins. They are essential for the precise segregation of materials during cell division. Chromosomes are composed of the DNA and associated proteins such as the histones. They represent the most condensed and visible form of genetic material in the nucleus. What about chromatids? Chromatids are found within chromosomes, specifically during cell division. They are two identical copies of chromosomes that are connected at the centromere. So chromatids are involved in accurate distribution of genetic material to daughter cells during cell division. Each chromatid contains a complete copy of DNA molecules. So chromatids are the result of DNA replication, where the original DNA strand is duplicated to ensure that each daughter cell receives a full set of genetic information. So what are the human chromosomes? Where are they? Human, typically they have 46 chromosomes in their somatic cells that are organized into 23 pairs. These pairs consist of one chromosome inherited from each parent. So the chromosomes are categorized into two groups. We have autosomes and the sex chromosomes. The autosomes, which is also referred to as the non-sex chromosomes, they are 22 pairs of autosomes numbered from number 1 to number 22. They are responsible for determining most of the individual traits and characteristics. While sex chromosomes, there is one pair of sex chromosomes which determine an individual's biological sex. So females have 2X, that is XX, whereas males have 1X and 1Y. Okay, so we have the XY chromosome. You can see chromosome 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. And then we have the 23rd is sex chromosome. Okay, you could have XX or you could have XY. So humans have 46 chromosomes with 22 pairs of autosomes and one pair of sex chromosomes, resulting in a total of 23 pairs. So the unique combination of this chromosome in each genome contributes to the genetic makeup and influences variability. What are mutations in human genetics? This refers to changes or alterations in the DNA sequence of an individual's genes. These alterations can occur naturally as a result of errors during DNA replication or environmental factors, or they can be induced intentionally in the laboratory settings. Mutations have a wide range of effects, from having no discernible impact to causing genetic disorders or contributing to the evolution of species. Mutations are a natural part of genetic variation and play a significant role in both disease and evolution. So while some mutations may be harmless or even beneficial, others have detrimental effects on health of a person. Types of mutation, we have point mutations. These are small scale mutations that normally involve a small change in single DNA base pair. There are three types of this point mutation. Could have substitution, where one base pair is replaced by another, or insertion, where one or more base pairs are added to the DNA sequence, or deletion, where one or more pairs are removed from the DNA sequence. We also have the frame shift mutation. These are just mutations that occur when there is insertion or deletion of base pairs that normally disrupts the reading frame of a gene, leading to a shift in the way the genetic code is read. They often result into non-functional proteins. For chromosome mutations, these mutations involve changes in either structure or the number of chromosomes. Examples could include deletions, where a portion of a chromosome is missing, or duplication, a portion of a chromosome is duplicated, or inversion, where a portion of a chromosome is reversed. A segment of one chromosome is normally moved to another chromosome in translocations. What are the causes of mutation? As we have already said, is that we could have spontaneous mutations and induced mutations. Spontaneous mutations occur naturally due to errors in DNA replication, spontaneous chemical changes, or exposure to environmental factors like radiation or other certain chemicals or drugs. Whereas induced mutation. These are mutations that are deliberately induced in a controlled laboratory setting. This could be for the purposes of research, okay, such as studying gene function or effects of specific mutations. Impact of mutation. We could have neutral mutations, beneficial mutations, and these beneficial mutations, which are rare, could provide an advantage where we have like resistance to diseases or improved traits. And harmful mutations, which are very detrimental and can lead to genetic disorders or diseases. These mutations normally disrupt the normal functioning of genes and proteins, leading to health problems. Genetic disorders, mutations are common underlying causes of these genetic disorders. Disorders such as cystic fibrosis, sickle cell anemia, the Huntington's disease. These disorders normally result from specific mutations. Genetic diversity. Mutation can also be essential for genetic diversity within population and contribute to the evolution process. They introduce variations upon which natural selection can act, leading to adaptations over generations. So we have single gene defectives and polymorphic gene defectives. So the single gene defective genes, these are thus the Mendelian or the monogenic disorders that are responsible for a wide range of inherited genetic diseases. So disorders that are caused by mutations in a single gene, and the inheritance pattern typically follows the Mendelian genetics, which includes either autosomal dominant, autosomal recessive, or the X-linked inheritance. Conditions could include cystic fibrosis, Huntington disease, Duchenne muscular dystrophy. We also have sickle cell anemia, hemophilia, phenylketonuria, polycystic kidney disease, the Church-Child disease, the Marfan syndrome, and the familial hypercholesterolemia. What about the polygenic and multifactorial disorders? These are the complex diseases and traits influenced by multiple genes as well as the environmental factors. So the polymorphic disorders result from combined effects of variation in multiple genes, such as contributing a small part to the overall size. Conditions such as type 2 diabetes. Of course, while we agree that genetics play a role in type 2 diabetics, is a complex polymorphic disorder. So multiple genes contribute to an individual's susceptibility to this disease, and these genetic factors interact with lifestyle, environmental factors such as diet and physical activity. The other polygenic or multifactorial disorders include heart diseases, obesity, autism, schizophrenia, height, cancer, Alzheimer's disease, and autoimmune disorders. So having those disorders, how can we manipulate the genome so that we fail to have those disorders? So that's why we have manipulating the human genome. So this involves making intentional changes to the DNA sequence of an individual's genes. So this field of genetic engineering has been advanced significantly in recent years, primarily due to the technologies like the CRISPR-Cas9. So we have gene editing technologies. So the most prominent method of manipulating human gene is the CRISPR-Cas9. And it's a revolutionary gene editing tool that allows scientists to target specific genes, make precise modification to the DNA sequence. It involves guide RNA, where these are designed RNA molecule guiding the enzymes to the target gene. Then you have the Cas enzyme, which acts as molecular incisors, okay, the scissor for cutting the DNA at the desired location. Then you have the repair mechanism, where the cell's natural repair mechanism, such as the non-homologous end joining or homology-directed repair, are used to introduce the changes to the DNA. So the editing will involve the guide, the Cas enzyme, and the repair mechanism. We can apply genetic editing in a number of ways, where we can use in treating genetic disorders, okay, treating genetic disorders such as cystic fibrosis, sickle cell anemia, and muscular dystrophy. We also have cancer therapy, where researchers are trying to explore gene editing to target and disable genes that are responsible for cancer growth. It's also important in drug development, agriculture, to engineer crops and livestock for improved yield, disease resistance, and nutritional content, and biotechnology, which is important in producing biofuel, pharmaceuticals, and industrial enzymes. We have ethical consideration in this case, guys. Gene editing in humans has raised a number of ethical concerns, particularly when it comes to the germline editing, the modifying genes that can be passed on to the future generation. So many countries have imposed restriction on the germline editing due to the potential of unintended consequences and ethical dilemmas. We have the possibility of the designer babies, where parents can select specific traits for their children, and this raises ethical questions about the commodification of human life. So we need regular oversight, where many countries have established regular regulations and guidelines on gene editing, especially in clinical application. So this regulation aims to ensure safety, ethical standards, and informed consent. So for the field of possibility, we find that the field of gene editing is continually evolving, and scientists are researching ways to make the technology more precise, efficient, and safe. There is a potential future application which may include in treating wide disorders like Alzheimer's disease, HIV, and advancing personalized medication. So manipulating the human genome through the gene editing technologies like the CRISPR-Cas9 holds immense promise for treating genetic diseases and advancing scientific understanding. It's important to know that although this is possible, it raises important ethical and regulatory challenges that require careful consideration and oversight. So how can you detect this abnormality? We have the preconception carrier screening. This is normally done before pregnancy or early in pregnancy, where you identify carrier status for specific genetic disorders to assess the risk of passing them to your offspring. We could also have first trimester combined screening, and first trimester is between that period will be between 11 to 14 weeks, where you assess the risk of Down syndrome and other chromosomal abnormality based on maternal age, ultrasound, and blood tests. This provides a risk score. We also have the cell-free DNA, and this can be done as early as 9 to 10 weeks of pregnancy, where you screen for common chromosomal abnormalities, especially Down syndrome that is very common, by analyzing fetal DNA in the mother's blood, and this normally provides high accuracy. We also have chronic villus sampling, CVS, and it's typically performed between 10th to the 13th week, okay? And it's kind of an invasive procedure to directly sample placental tissue for chromosomal abnormality and genetic disorders. This one normally provides definitive results. We have amniocentesis, and this is typically performed between 15 to 20th week of gestation, and is an invasive procedure that collects and analyzes amniotic fluid for chromosomal abnormality and genetic disorder, and it provides definitive results. So this is for the prenatal genetic testing. What about the newborn genetic screening shortly after birth? So we have the newborn screening that is normally done within first days of life. You screen for a panel of genetic and metabolic disorders, typically using a blood sample. Early detection allows timely treatment. We have genetic testing in adulthood. This is done throughout the life based on the clinical indication. So we have genetic counseling and testing where it can be done at any age based on the family history and clinical indication. It provides genetic counseling to assess risk of inherited conditions and offers genetic testing as needed. We also have diagnostic genetic testing performed at all ages when a specific condition is suspected, and it analyzes the DNA to diagnose genetic disorders based on the clinical symptoms and family history. We have carrier testing, and it's typically done before or during family planning. So we have the purpose of this one is identifies carrier status for specific genetic conditions to assess the risk of passing them to their offspring. Lastly, we have cancer genetic testing, and this is typically for individuals with family history of cancer, where you screen for genetic mutation associated with increased risk of cancer to guide prevention and treatment decision. So guys, where are we? We are done with describing cell specialization, describe the four body tissues. We have described process of cell-to-cell communication. We have outlined the human genetics and some of the disorders that are common. We can end with induction process of a cell. So what is the induction process of a cell? Cell induction is a process by which a group of cells influences the fate and behavior of neighboring cells, often causing them to differentiate into a specific type of cells or undergo particular developmental processes. This phenomena plays a crucial role in embryonic development and tissue regeneration in humans. So when starting with the embryonic development, you find that during embryogenesis, cell induction is normally responsible for organization of various tissues or organs. So we have specialized signaling cells, we call them the morphogenes, are secreted by one group of cells and diffuse to nearby cells triggering specific responses. These responses include changes in gene expression and cell differentiation, leading to development of diverse cell type and structures. We have stem cell differentiation. So induction can also occur in adult tissues, particularly in the context of stem cell biology. So stem cells have the potential to differentiate into various cell types, and their fate can be influenced by signals from their microenvironment. These cells can include growth factors and cytokines released by neighboring cells, which guide the differentiation of the stem cells into specific stem cell lineages. Cell induction is also essential for tissue repair and regeneration process in adults. So after injury or damage, surrounding cells and the extracellular matrix normally release signals that induce nearby cells, including those stem cells, to proliferate and differentiate to replace the damaged cells. So clinical application is that understanding of cell induction process has significant clinical implication. Researchers are exploring ways to harness cell induction for therapeutic purposes, such as regenerating damaged tissues, treating degenerative diseases, and enhancing wound healing process. So cell induction in humans involves the influence of one group of cells. Okay, they influence the fate and behavior of neighboring cells. This leads to differentiation into specific types of cell and development processes. So this process is crucial for embryological development, tissue repair and regeneration, and clinical manifestation. Thank you guys. We can look at the review questions. So as you wind up the lecture, you need to be able to outline the different types of cells for the epithelial, connective, muscular, nerve. You need to describe the functions of the epithelial tissues, explain the process involved in cell-to-cell communication, define genes and describe human genomes, and lastly, you need to be able to identify the single defective genes and polygenic disorders. Thank you so much, and that is the principles of human genetics. Remember to access the free quiz in the description section testing all the 5 objectives.